So I was sitting in bio class, listening to the endless droll of my teacher, when I heard a few words that caught my attention. Alter DNA. And that's when the spark went off in my head. You see, a few weeks ago, I heard an NPR about an enzyme that could alter DNA. Yeah, that double helix thing that life is kind of based on. They talked about how it was life-changing and beyond revolutionary. And just like in class, I remember to start to pay attention. Because honestly, to a high school student, NPR is very similar to a babbling teacher. Although, altering DNA is not some daily news filler. It's a significant innovation in genetics. Messing with nature is not a new thing to humans. We've developed artificial selection, genetic selection, and cloning. The earliest humans cultivated crops by choosing the highest yielding plants to regrow in the next season. Gregor Mendel crossbred species to extract the best traits of each one. And cloning has revolutionized genetics in the modern era with Dolly. We've understood DNA since the mid-20th century and shook the forces of nature with science. However, each of these genetic advancements has affected the DNA of the next generation of species. And that's where this enzyme makes its mark. This discovery was all natural. It's found in bacteria, but more importantly, could alter the DNA of living creatures and cells, which means your parents, grandparents, and you could change your lives at any time. Well, it's still kind of being worked on, but I'll talk about that later. Nevertheless, this is a major breakthrough in the sciences, and they call this spectacular enzyme Crispr, but spelled without the E. Crispr, or Cas9, was originally found in bacteria as early as 1987, but were only found in the form of regular repeats in DNA of bacteria, which scientists only named SRSR, or short, regularly spaced repeats. Yeah, I know, whoever came up with that name is a true creative genius. CRISPR is much easier to say and work with, but SRSR won't be renamed CRISPR until 2002, past those mysterious times our parents like to call the 90s, and some of us briefly lived in. In truth, the enzyme is named Cas9, and the SRS sequence in DNA is the CRISPR part. For clarification purposes, though, we'll use CRISPR for the process as a whole throughout the rest of the video. Scientists investigated SRSR and found that special enzymes were being created from an SRSR gene. These enzymes were able to wrap around DNA and, most surprisingly, cut it. Those new enzymes had another unique trait in addition to be able to cut DNA. The SRSR gene created bits of virus DNA and inserted it as a spacer in the enzyme. Those spacers acted as a wanted poster for the enzyme to read, track down, and demolish. It was finally resolved in 2005 that these enzymes and the SRSR gene were one of the best defenses against viruses bacteria had. But before I go into the modern uses of CRISPR, it's important to understand how CRISPR works in bacteria and cells. The CRISPR enzyme is shaped kind of like a U, and the shape allows the spacer to be stored in the cutout of the enzyme. So we will represent CRISPR as a Pac-Man. But instead of eating red cherries, CRISPR's on the hunt for dangerous pathogens. You see, those virus DNA spacers were actually a copy of a virus which had previously attacked the bacteria. When an unknown virus attacked the cell, and in the odd circumstance that the bacteria triumphed, the viral DNA would be captured and inserted as a spacer into the Cas9 enzyme. CRISPR will surround a DNA strand and use the properties of a helicase enzyme to split the DNA into two strands. Helicase enzymes are an important part of life, which all cells contain, and are primarily used to unravel the DNA. Helicase enzymes do this by breaking the hydrogen bonds holding the amino acids together. From there, one of the strands from the spacer in the Cas9 enzyme, which is identical to foreign viruses, tries to attach to the newly separated DNA by bonding to the unpaired amino acids. And if it's a match, then CRISPR goes on attack. This is when the cutting ability of CRISPR is on display. CRISPR has the properties of a nuclease enzyme, which specializes at cutting nucleic acid backbones, which reside in DNA. It does this by using a hydration reaction to break the phospheter bond, which hold the phosphoric acid and pentose, the main two components of the DNA backbone, together. By splitting the invasive DNA, CRISPR eliminates the invader's functionality. The takeaway from this discovery was that cells were learning from their previous victories in order to get the upper hand in the next attack, just like our immune systems do. Once scientists realized what CRISPR was actually doing, the enormous importance of its future capabilities were just starting to be unraveled. Scientists thought that they could insert CRISPR into organisms with faulty DNA and snip the incorrect DNA. The faulty DNA would act as a spacer, so CRISPR would only target the DNA with problems. Pain and suffering solved, right? Well, no, not exactly. As in most science, it's not that simple. Humans need functioning DNA to survive, and if CRISPR is just going around cutting our life code, then we're more doomed than before. For CRISPR to work for bacteria, the viral DNA can't rejoin, or else the virus can go back to terrorizing the bacteria. Amino acids need to be correctly linked back up in the DNA and have to be linked in a specific pattern. The complex process needed to reform the DNA makes the split DNA very unstable and unable to ever rejoin. The difficult process aids bacteria in its fight against viruses, but was troubling for scientists. Not only did they have to link the DNA back up, but when it rejoined, it had to be different from the original and had to fix the problems of the previous DNA. Scientists decided to insert a portion of the correct DNA to act as a bridge between the two parts of the cut DNA and fit perfectly into the unbonded amino acids. This process repaired the old DNA and created the correct DNA from its predecessor. From there on out, it was possible to edit DNA into any strand as long as Cas9 enzyme with the correct spacer in it and a repairing section of DNA were inserted together. CRISPR opens up the opportunities to fix essentially every DNA mutation. That means no more sickle cells, genetic factors to cancer, Alzheimer's, Angelman's, and much, much more. The world's not going to be the same place after CRISPR becomes widespread. But it might take a little while until CRISPR becomes an everyday reality. Chinese scientists have used CRISPR to edit the genomes of human embryos. However, while they completed their intended task, their success rate was very low. That doesn't mean it can't work, though. Similar tests on sheep, mice, and pigs have all yielded amazing success. CRISPR is still in the very early stages of development, and it's predicted to revolutionize genetics. The genome of an elephant can be replaced to mirror that of a woolly mammoth, allowing us to bring back a wide array of extinct species. Human genetics could also be essentially chosen to contain good traits and not contain any diseases or unfavorable mutations. But CRISPR isn't guaranteed either. Sure, it'll be perfected after multiple studies and tests, but there's a moral view on genetic tampering. Cloning was the big deal in genetics during the 90s, and it's still debated over whether it's morally right to do with humans. CRISPR is on a whole new level, though, and I can't imagine the struggle our population will have with disputing the moral and scientific values of using CRISPR or not. Although test tube babies have become accepted, and almost 1.5% of newborns now originated from test tubes. If something like test tube babies can overcome the ethical friction, then there's no reason CRISPR can't. Nevertheless, CRISPR will be opposed and questioned for decades, and I can't tell you what will be the outcome. But I can tell you one thing. My generation will be the deciders of what will happen to CRISPR, and now is the time for CRISPR to be studied, debated, and researched, so we can make the right choice down the road.